There should be 3 distinct layers in the shop. Pour Kahlua into a... Pour Kahlua into a...
In order for your body to produce proteins, it needs to utilize a molecule called RNA. Notably, this has a similar name to DNA, i.e. 3 letters ending in Na. However, the difference is the first letter of this is an R, and this R stands for ribose, hence ribose nucleic acids, as opposed to deoxyribose nucleic acids. And these are heavily involved in protein synthesis. Below is the structure of an RNA nucleotide. You can see that there is a phosphate group, a pentose sugar, notably ribose, not deoxyribose, and an organic base. One of the main things you'll have to remember about the difference between RNA and DNA is that the bases are different. For example, where there would be a T in DNA, there is a U in RNA, and I'll explain this later on. In all, there are 3 different types of RNA. There are messenger RNA, depicted at the top here, there's tRNA, depicted at the bottom, and there's also ribosomal RNA, which you don't need to know about for this course. mRNA stands for messenger RNA. This essentially provides a blueprint for protein structure. We know from Unit 2, I think, that the triplet code codes for proteins. For example, this is a triplet, this is a triplet, and these code for a specific amino acid in protein structure. So you can say that messenger RNA is the template for DNA synthesis. tRNA stands for transfer RNA. Notably that the structure of this is it's very different. It's more of a clover-leaf shaped structure. Down here is the anticodon loop, which is complementary to a codon on the mRNA molecule. This is important to DNA RNA translation, which is when proteins are produced. At the top of the tRNA molecule there is a site of attachment for an amino acid. You don't need to know this for the course, but I thought it was interesting that this was an ester bond, which connects it to the amino acid, which leaves the amino acid with a positive charge. The process by which mRNA is produced is called RNA transcription. This is carried out by the enzyme RNA polymerase with additional help from DNA helicase for a specific section. At this point, it's important to note that DNA is double-stranded. Now this has benefits in terms of stability of the molecule and therefore ability to be passed down from generation to generation. However, this also leaves the problem that one of these strands doesn't code for DNA, which isn't really a problem per se, but it means it isn't necessary for this process. So this top strand here is the non-coding strand, and the bottom strand is what we call the template strand. So to begin RNA transcription, DNA helicase acts on a section of DNA about 12 bases long or so, exposing the nucleotides. Now RNA polymerase bonds are the start codon for this, and starts transcribing the DNA. The way this happens is that free nucleotides from the nucleus flow into their complementary bases inside RNA polymerase, and RNA polymerase joins the phosphate sugar backbone, forming a pre-mRNA molecule, which then flows out through the nuclear pore, or a nuclear pore rather. Now behind the RNA polymerase molecule, the hydrogen bonds joining the DNA molecule reform, so only a few bases are exposed at any one time. As I mentioned earlier, the base uracil replaces thymine in RNA, which is shown here when U binds to A. This is the stage where this happens. Splicing is the second stage of mRNA synthesis. This is the conversion of pre-mRNA into full-blown mRNA. Now this is essentially taking the introns out of the pre-mRNA molecule. You should know from Unit 2 that DNA consists of introns, non-coding DNA, and exons, coding DNA. I always thought that these were badly named because exon sounds like it should have been left out and introns sound like they should have been left in, but I don't know who decided that. So with splicing, essentially the introns get taken out, so you have a molecule which is just exons, or all coding DNA, which can now be used in protein synthesis or translation. DNA translation is the final stage of protein synthesis. This is facilitated by ribosomes. The first stage of this is the small unit of the ribosome forms a complex with the mRNA template. The large subunit and a complementary transfer RNA molecule with a complementary anticodon. Now it's easier to explain that this is called the anticodon because it's the opposite of the codon here. They are complementary to each other. So we've got the large subunit here. There are 3 binding sites in the large subunit. There's the P site, which is where the initial transfer RNA binds, the A site, where another tRNA molecule can bind, and the E site, where the tRNA molecule can exit after it's passed on its amino acid. So at this stage, another tRNA molecule with another amino acid can come along. This tRNA molecule flows into the A site of the ribosome, forming again hydrogen bonds with the complementary bases. The ribosome then forms a bond between the 2 molecules, forming a peptide bond. The ribosome then moves along the mRNA strand by 1 codon, leaving the first transfer RNA in the E site, so this leaves the ribosome and floats back off into the pool to collect another amino acid of its complementary structure. This then leaves the A site free for another amino acid to flow into and bind with its complementary codon. And again, the ribosome forms a peptide bond between 2 amino acids. This whole process repeats itself over and over in a phase called elongation because the protein chain's getting longer, until the ribosome reaches a stop codon. So when the ribosome reaches this, it recognizes it as a stop codon and dissociates the large subunit, small unit, transfer RNA and the protein molecule. And the outcome of this is that you have a brand new protein molecule fresh from translation.